Okay, so what about animals? Okay, they don't have the ability to become everything, all types of cells. All right, so we can use that in order to mess with their genes, basically. And what I'm really talking about is cloning. So it's called nuclear transplantation, and basically what you're doing is you're taking an unfertilized egg, you take out its nucleus, and then you put the nucleus of a fertilized egg into that cell and see how it develops. All right, does it develop normally? Well, in the case of the clone cell, then it does develop normally. All right, given that it's a young cell, the donated DNA is very young, then it's okay. So it replaces, basically that nucleus is going to replace the nucleus of the unfertilized egg, and that's what we call clones. All right, because the DNA inside those cells is not from the original mom. Okay, it's from an outside source. So, first, it shows that nuclei do change as the cell differentiates. Secondly, that the nuclear potency decreases as cell divisions increase. Okay, so as the cell starts to get more differentiated, it divides more often, then the nucleus doesn't have as many types of totipotent cells that it can become. And then thirdly, cells differ because they express different proteins in a specific tissue. Okay, so every cell that we have is going to have all of your genome in it. Okay, every single cell will have every single piece of DNA sequence that it needs to become anything. Does it? No, but it has all of it there. So what is making it into a heart cell? What's making it into an organ? All right, what's making it into a specific organ? Well, it's different portions of that DNA that's going to be expressed. So really what it is it's transcriptional regulation. And transcriptional regulation is going to determine what type of what type of tissue a specific cell is going to be via the process of determination. Okay, so it's a process that leads to the observable differentiation of a cell, where a cell is going to be irreversibly committed to a specific tissue fate. Okay, so these cells are going to make tissue-specific proteins. They're going to start expressing tissue-specific proteins that are characteristic of only that tissue to do only that job. All right, so that's where it becomes irreversibly committed. Now, the example they give you in the book is from muscle determination, okay, muscle cell formation. Now, muscles are going to be, the cells of them are going to be determined to be muscles by something called a master regulatory gene. And all of your tissues are going to have these. All right, it's a series, it's usually a couple of genes that are working in coordination that are going to basically commit a cell to its fate by expressing certain genes in it. Now, it's called MyoD in the case of the myoblast cells. So what's going to trigger the direction of this master regulatory gene to start committing these cells? Well, there are two sources of trigger. All right, the first one is going to be maternal cells, or the maternal RNA and the protein molecules that are in the cytoplasm of the unfertilized egg. And then the second is going to be the external environment. All right. Now the first one I'm talking about is going to be the cytoplasm of the unfertilized egg. So look on page 412. All right, you'll see that in figure 21.1 part A, the cytoplasm is going to have, it's going to be a heterogeneous mix, okay? So it's not going to be like the organelles and the proteins are all spatially arranged evenly in the cytoplasm, right? They're going to be parts that have more concentration of certain things than the other parts, all right? So as soon as fertilization occurs, the cell nuclei are going to divide, right? They're going to have two cells now. But now the nuclei are going to be in different cytoplasmic environments, all right? And these are called cytoplasmic determinants because they regulate the expression of genes affecting the developmental role of specific cells. Okay, so the cytoplasm, what's in the cytoplasm is going to determine the role of those cells. The second trigger is going to be external environments. Now, The external environment is going to become more important as the cell divisions occur, all right, as the cell numbers increase. All right, so signaling between cells and signaling molecules from nearby cells are going to elicit changes in their specific target cells, and this is called induction. We kind of talked about cell signaling when we talked about it in chapter 11 or whatever it was. But basically, cells are going to secrete signaling molecules that will be received by specific target cells, and it's going to help determine their fate as well. So we're going to see this. We're going to see this type of pattern formation. All right, basically what that means is a spatial organization of all of these cells, because these cells are going to divide. They're going to be determined to be a specific tissue, and that tissue is going to move to be in a specific place in your body. All right, so we're looking at pattern formation. How are these tissues and organs going to be spatially arranged to create this organism that has axes and different ends? Okay, so in animals, that always begins in the early embryo. It's usually done by the time the embryo hatches or the baby's born. All right, so this positional information is going to tell a cell its location. Okay. And so we'll see it, we see it very well in the life cycle of Drosophila. All right, so I'm on page 413. This is chapter 21, this is figure 21.11. All right, and we're looking at the key developmental events in the life cycle of Drosophila that show you where the three regions, the head, the thorax, and the abdomen, are going to be. Okay, so it has an anterior and posterior position, an axis, and it has a dorsal ventral axis. Okay, so in the first step, the fertilization is going to be followed by rapid cell division, but this division is different. Okay, there's no gap 1 or gap 2 phases. It's just going through synthesis and mitotic phase. No cytokinesis. So you see that you're going to have this multinucleated cell, one cell with many nuclei in it. And then at division number 10, okay, the nuclei are going to start to move to the outside line of the early blastula, okay? And then after that cell, at mitotic division number 13, it's going to start to undergo cytokinesis so that all of the nuclei are separated by a plasma membrane, okay? All right, segmentation is going to start to begin, okay? It'll undergo 3 larval stages. It'll undergo a pupa stage, and then it'll go through metamorphosis into an adult fly. Okay, so what genes then are going to be regulating all this and directing all of this morphogenesis? Okay, so basically, it's hard to look at developmental aspects of an embryo by looking at a normal developing embryo. So we look at mutants. Okay, what genes affect what proteins, affect what parts of development? We look at mutants to try and find this. Now, the caveat to looking at mutants is that, the pro of looking at mutants is that you can trace back what gene went wrong to kind of put it with a specific protein deficit or enzyme deficit or whatever. So abnormalities always lead back to some sort of genetic problem, and we can trace it back to that. But in the case of mutants, then, sometimes you get what's called an embryonic lethal, where the mutation is so severe and it happens so early in development that development of the embryo would not be able to survive without that good gene. And so the embryo dies, basically, before it even starts to develop. And so we can't look at just pure homozygous mutations. We have to look at carriers, because in order to get around the embryonic lethal problem, we have to look at the carriers of it, especially if it's a recessive mutation. So we look at carriers. So we ask ourselves now, okay, how does her maternal molecules and her maternal mRNA and her cytoplasmic determinants play a role in axis formation, in pattern formation, in all this stuff? Okay, so let's go to page 414. Okay, so When we talk about cytoplasmic determinants, which remember are these things in the mom's unfertilized egg that are going in the cytoplasm of that unfertilized egg that are going to help determine what the development of the embryo is going to look like. Cytoplasmic determinants have determined axes, okay, plural, axes. They're encoded by genes in the mother called maternal effect genes, all right? If these are mutated in mom, they're mutated in the child, right, or in the embryo, in the offspring, right? They also call them egg polarity genes because they actually affect the orientation of specific ends, so the anterior posterior end, so the polars of the egg. Okay, now, how do we decide what's anterior, what's posterior? All of that is determined by some sort of concentration gradient of a protein that is going to be secreted. Okay, so the great example that they give you in the book is going to be bicoid. All right, the bicoid gene is going to determine the anterior and posterior end. Basically, it's a protein that is called a morphogen. Right, it's a type of protein that's going to have something to do with the morphosis of, or the morphogenesis of an organism. Okay, so they call it a morphogen. Now, it's a gradient, which means that the anterior end of this cell, all right, is called the anterior end because it has a high concentration of bicoid protein. All right, and then as the protein diffuses across the cell, the posterior end is not going to have as much, so it's going to have a smaller concentration or a lower concentration of bicoid protein. All right, so that determines that the high end bicoid protein is going to be the anterior end. The low end of bicoid protein is going to be the posterior end. Okay, so this is what's going to create segmentation as well. All right, so you've got these maternal effect genes that are going to affect the axes, posterior, anterior, dorsal, ventral. And then you've got what are known as these segmentation genes, which are going to start to segment out, maybe in the case of the Drosophila fly, it's going to be the head, thorax, and abdomen region. All right, they're called gap genes because these are the basic subdivisions of anterior and posterior. Okay, you've got the pair rule genes, which means you're going to have pairs of segments on each side because the Drosophila fly has bilateral symmetry. And then you're going to have something called segment polarity genes, which means that you are going to give an anterior or posterior end of each segment. Okay, so you have the overall anterior, posterior, head and butt region. And then you'll have, for each head, you'll have the front part of the head and the back part of the head. Okay, so that's polarity of each segment. All right, now we're going to go into how do you determine what cell becomes which body part. And this is from the homeotic genes. Now they give you a really good hierarchy of gene activity on page 417. It's good to start to map out these types of genes and how they're going to be distributed. All right, so it's like a cascade almost of genes. All right, so you start with the maternal effects, you go to the segmentation, then you go to starting to ID body parts, and then you're going to start doing other regulatory genes of the embryo. Okay, so we're on homeotic genes of the embryo. And these are genes that are going to direct the identification of specific body parts. All right, U5 cells are going to become the antenna. U5 cells are going to become the eye. All right, that kind of stuff. All right, now, if you look up on page 21.1 or on page 417, figure 21.14, you'll see that homeotic mutations are going to give you abnormal patterns of formation. All right, so in the case of the normal one, which is on your left, you have the eyes and then the antennas are in front of the eyes. And in a mutant, all right, this is a mutated homeotic gene. It has distributed cells to become legs where the antenna should be. Okay, so it's still a functioning embryo because it doesn't need antenna for viability, just to stay alive. But now it's got legs there instead. It's kind of gross to look at, but I mean, that's what's going on. Okay, so we're going to talk about, in the next video, I'm going to talk about homeobox genes.